So today's journal club discussion is about the dynamic enhancer landscapes in human craniofacial development. So this was published in the Journal of Nature Communications in the year 2024 March. So before moving on to the novelty and methodology section, there are certain terms that we need to know which is being used in this study. So craniofacial development, we all know, it is a process by which the face in the skull develop during the embryonic growth. So it involves many genes and regulatory elements that control how different tissues grow and connect. So what are enhancers? So enhancers are like the switches which I told earlier during the seminar session to turn genes on and off at the right time and place. So these enhancers do not code for protein but help control when and where specific genes are active. And there is something known as the histone modification. It is nothing but a chemical marker which is found on the histones. So they are proteins that help pack DNA. So if region of DNA has this marker, it means the genes in that region are likely to be active and being used by the cell. So that is the histone modification. So, and something that is known as the attack ATAC sequence, assay for transposase accessible chromatin with sequencing. So it is a method used to identify open regions in the genome where proteins like the transcription factors can bind. So open chromatin regions are where enhancers and other regulatory elements are being active. And another one is the chip sequencing, chromatin immunoprecipitation sequencing. So it is a technique used to find which part of the DNA have specific protein markers such as the histone modification protein. So it helps scientists identify active enhancers and understand how the gene regulation actually works. Next is the RNA sequencing. So this is a method that measures which genes are active in a cell at a given time by sequencing the RNA molecules. This helps link the enhancers to the genes that they regulate. Next is the single cell RNA sequencing. This is a technique that allows researchers to study gene activity in individual cells rather than averaging results across many cells. So this provides a detailed map of which genes are active in different cell types. We all know this is single cell RNA sequencing. There is something known as a single nucleus attack sequencing which is performed on individual cell nuclei. So it helps identify which regions of DNA are open and potentially active in specific cell types. Next is the epigenomics. Epigenomics, we all know, is a study of changes in gene activity that do not involve changes in the DNA sequence itself. So it includes modifications like the examples of the epigenetic modifications are the histone modification and the ATAC sequencing. So gene expression is the process by which genetic information in DNA is converted into the functional products such as proteins or the RNA. And in this particular study, they used something known as the scan phase X, which is nothing but a single cell gene expression data set created in this study that maps which genes are active in different cell types during the craniofacial development. Then there is something called as a scan phase N. So it is a database of craniofacial enhancers that provides information on which enhancers are active in different cell types. And they use the transgenic mouse model, a special type of mouse that has been genetically modified. Some genes have been added. So they have genetically modified to contain the human DNA sequencing. Next is the lac-Z reporter assay, a technique used in transgenic mice where an enhancer is linked to a reporter gene, lac-Z, which provides a blue color in cells where enhancer is active. Emissary reporter it gives red color when the gene is active. So this is the basic terms which is being used. And the comparative genomics is a method that compares genomes of different species like the humans, mice and chimpanzees. So in this study they have used the comparative genomics, and there is something called as evolutionary conservation where when a DNA sequence remains similar across different species, it suggests that the sequence has an important functions to be there. Over the evolutionary period, this particular DNA sequence has played an important role. Then next we have the genome-wide association studies, a method used to find genetic differences that are linked to diseases or traits. And cell type specific enhancers, that is enhancers that are only active in certain type of cells such as bone forming cells or skin cells. So in this study, the novelty is that understanding the birth defects. Some people are just born with facial malformations, like the cleft lip or the cleft palate due to the genetic changes. So what they have done is study is to understand what part of the DNA may be involved in these conditions. So creating a genetic map of facial development and finding human specific enhancers. So which part exists only in humans, meaning this could be responsible for the particular facial feature of humans, and connecting genes to the facial growth. By identifying enhancers and genes they control, scientists can figure out which part of the how different parts of the face like the jaw, nose and the palate develop. So this is what they are about to do. And the methodology which they used was they collected data from human embryos. So the researchers studied face tissues from human embryos at different stages of development, 7 weeks to 8 weeks, 7th and the 8th week. So they looked at which part of the DNA were active during this time to find the enhancers. So step 2 is identifying the active enhancers. So to find enhancers, they use the techniques which I have told earlier. This is ATAC sequencing, which finds open DNA regions, chip sequencing to find specific histone markers where the enhancers will be active, and the RNA sequencing, which measures which genes are turned on at different stages of the face development. By combining these methods, they created a list of nearly 14,000 enhancers that might be important for the craniofacial development. And finding out where enhancers work in the face is the step 3. So they use the single cell RNA sequencing and the ATAC sequencing to check which enhancers are active in different cell types in the face. So this helped them figure out which enhancers control the formation of the skin, bone, cartilage in the face. So testing enhancers in mice. Since human embryos are difficult to study, the researchers used the transgenic mice model in which the human DNA sequences are inserted into their genome. They attach reporter genes, that is the LAC-Z and M-cherry, to find out the particular enhancer which is active, that will be showing in the color change, that is it turns red or blue, that particular site. And this experiment proved that many enhancers were real and active in the development of the facial features. So next what they did was they compared the human enhancers to the other species. So like the chimpanzee, mice and the chickens, some enhancers were shared across different species but others were only found in humans. So they proved that these genes were only responsible for the formation of the facial features of humans. So moving on to the results, a total of 13,983 craniofacial enhancers were found only in human embryonic facial tissues. Some enhancers were active for a short time while others stayed active throughout development. Some enhancers are human specific, that is not all enhancers are found in the humans, were found in other species also. So some enhancers were unique to only us. And enhancers link birth defects. So the researchers found that many enhancers are located near genes linked to the craniofacial disorders like the cleft palate. So this suggests that the mutations in these enhancers could be responsible for the birth defects, facial birth defects. And they also developed a new atlas for the facial development. The study created the scan phase X and the scan phase N, which is they're all nothing but the enhancer maps to identify which are the enhancers or the active parts in the gene regulation. So these maps shows which enhancers and genes are active in the facial features. So this is being used as a tool to study the craniofacial development in more detail. So the limitation of the study was not all enhancers were fully verified. Even though some enhancers were tested in mice, not all of them were fully validated. So some might not be as important as we see them. So and there's also this challenge with the single cell data. Single cell techniques sometimes miss weakly expressed genes. So some enhancers may have overlooked or misclassified. So differences between humans and mice. The study used mice to test human enhancers, but mouse models aren't perfect. So some enhancers might work differently in actual human tissues also. So only studied early facial development. That is, in the methodology they have mentioned only the 7th and 8th week of the development has been tested. So later stages of the facial growth are missed. So that is one of the major limitations in this study. Thank you.